Hello, everyone, and welcome to today's webinar. How can liquid biopsy help in the management of patients with liver cancer? I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Agilent. To learn more, visit agilent.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Augusto Villanueva, Assistant Professor at Icahn School of Medicine at Mount Sinai, New York. Dr. Villanueva, you may now begin your presentation.
Thanks for the introduction and thanks for the organizers for inviting me to give this webinar on one of my main interests, which is the role of liquid biopsy for the management of liver cancer. These are my disclosures. I will include a discussion of one of our patents at the end of the presentation. So this is the outline of what I will discuss in the next 40 to 45 minutes. First, I will give you an introduction of what are the clinical needs for hepatocellular carcinoma, or HCC, which is the most frequent form of primary liver cancer. And after that, I will specifically discuss what I think will be the role of liquid biopsy for improving early detection approaches for this malignancy, and to derive biomarkers of treatment response, which is a clinical unmet need that hasn't been addressed properly for patients with hepatocellular carcinoma. So to start, a little bit of introduction of what liver cancer is. Here you have a summary of the estimated number of new cases and estimated number of new deaths per year in the different tumor types. So as you can see, the number one in terms of incidence and mortality is lung cancer, and liver cancer is around the fourth in terms of mortality. The size of the dot represents the lethality of malignancy. So the bigger the dot, the more lethal is that tumor type. For instance, pancreas is the most lethal malignancy, and liver cancer is the second most lethal tumor. We know that in terms of incidence and trends throughout the last decade, liver cancer has increased both in male and females. Here you have a summary of new cases of different tumor types in the United States between 2009 and 2013. It's a bit dated, but the more recent data is very similar. Despite liver cancer is not among the top in terms of actual number of cases, if you look at trends and changes in incidence, is the number one, or the tumor type that has increased the highest in terms of new cases for both male and female. Liver cancer has a number of singularities that makes it kind of a unique tumor compared to other solid malignancies. And one of them is that it's very rare to develop HCC, liver cancer, in a healthy liver. That's as opposed to colorectal cancer or breast cancer, where most patients have a healthy organ. In HCC, most patients have some sort of liver disease. The majority of them will have cirrhosis. Now, if you look worldwide, the prevalence of the incidence of HCC or liver cancer parallels the prevalence of incidence of chronic liver disease. For instance, here in Asia, East Asia, and Sub-Saharan Africa, you see a lot of red because a lot of cases of liver cancer associated mostly to infection with hepatitis B virus. China is one of the #1 countries in terms of incidence, and it's estimated that around 8 to 9% of the population is infected with hepatitis B. That's a huge risk factor for development of liver cancer. In Egypt, most of the patients have hepatitis C-related HCC, as 14% of the population is believed to be infected with this virus. The number one country in the world in terms of incidence is Mongolia, with 80 cases per 100,000 inhabitants, a very high incidence of liver cancer, one of the major causes of death in this country. In the US and in Western Europe, the estimated prevalence is around average is between 10 to 12 cases per 100,000 inhabitants. As I mentioned before, it's very rare to develop HCC in the lack of chronic liver disease. The main causes of liver cancer and as an extension of chronic liver disease are hepatitis B, hepatitis C, alcohol use disorder, and non-alcoholic fatty liver disease, which is increasing dramatically in Western countries. So when patients get any of these causes for chronic liver damage and they reach the stage of cirrhosis, the risk of HCC increases to 1 to 3% per year. Now, that is the rationale to recommend surveillance programs in these patients. So every cirrhotic patient should be enrolled in a surveillance program that includes an abdominal ultrasound and alpha-fetoprotein measurement every 6 months. There are some patients who can develop HCC before the stage of cirrhosis, and that poses a significant problem because since these patients are not enrolled in surveillance because the prevalence or the incidence of the tumor is very low in these patients, when they are diagnosed, the likelihood of these tumors being at later stage is higher. And that limits our possibility to cure these patients. As I mentioned before, the current recommendation for surveillance in HCC includes abdominal ultrasound and the quantification of serum alpha-fetoprotein every 6 months. And this has a sensitivity of 63% for the detection of early stage tumors based on this meta-analysis. It's interesting to see that alpha-fetoprotein, the only serum marker that is recommended for surveillance, is quite old. Actually, the first report dates back to 1970. So after 50 years, we've been unable to derive new biomarkers that are good enough to detect patients at early stages when they can be potentially cured. We have an additional singularity in HCC, is that to diagnose patients, we don't need a tissue biopsy. So a patient with cirrhosis that has the typical imaging features of nodules in the liver, so you have a CT scan of a cirrhotic liver, the patient has ascites, and you see these nodules in the liver. They are hypervascular in the arterial phase and they wash out in the venous phase. This is enough to diagnose this patient with hepatocellular carcinoma. So that's very convenient for patients because it facilitates things. But the downside is that this limits our access to tissue biopsies to run biomarker studies. And that has been an endemic problem in HCC research. So when you look at the clinical management of these patients, this is the BCLC staging algorithm that classifies patients in 5 different stages and provides a specific treatment recommendation for these patients. It's approved by the American and European guidelines for the clinical management of patients with hepatocellular carcinoma. Most of the changes that have happened the last 10 years in terms of the clinical management of these patients happen at patients with advanced stages. So 10 years ago, or more than 10 years ago, in 2008 or 2007, the first systemic therapy was approved for hepatocellular carcinoma, it's seraph. And after 10 years of negative trials, in the last 5 years, an additional 8 new compounds have been approved by the FDA for the management of these patients. And they're listed here. Actually, some of these drugs are now being tested at earlier stages. Now, the problem is that the majority of the trials that were testing these compounds were running parallel. There's a little confusion on which is the best sequence of therapy that patients should receive to maximize clinical efficacy and minimize potential toxicity. Since there are no biomarkers approved, this is kind of the question of, what is the first line best option? What's the second line? How patients should receive immunotherapy if they have received already, tyrosine kinase inhibitor. So there's a little bit of interest in developing tools or evidence that could guide physicians on the best way to allocate and sequence these therapies. There's only one biomarker that has been approved for the management of HCC, remember we talked about alpha-fetoprotein in the early phases of the disease, as it's also recommended for early detection. Well, there were 2 clinical trials that showed that patients with advanced stage HCC with high AFP levels, they do well with ramucirumab, which is a monoclonal antibody. So this is the only instance where a biomarker can tell you if the patient will benefit significantly from a given intervention, a given drug, if the patient has this biomarker. In this case, was high levels of AFP more than 400. The field, or let's say the management of HCC changed significantly last year. So this is the results of the randomized phase 3 trial that was testing a combination of atezolizumab plus bevacizumab versus sorafenib in first line for patients with advanced stage HCC. And the relevance of these studies is twofold. The first one is that for the first time after almost 12 years, you have a drug, in this case was a combination of atezolizumab plus bevacizumab, that outperformed sorafenib, was better, improved survival compared to sorafenib in patients with advanced stage. And that was until that trial, nobody was able to do that. And the second major thing about this study is that for the first time, it shows that a combination of, in this case, an immune checkpoint inhibitor and an anti-angiogenic drug is highly effective in patients with HCC. What is striking is when you look at response data, approximately around 30% of the patients treated with a combination will have an objective response as opposed to 10% of the treated with sorafenib. So now we have this combination therapy that is very effective in at least 30% of the patients, because we know that these 30% of the patients are the ones that will do relatively well over time. So this is a summary of the landscape of biomarker needs in HCC. We talk about risk of HCC development. So again, patients, for instance, with non-alcoholic fatty liver disease, they're at risk of developing HCC, but the risk is very, very small. Now, the number of patients with non-alcoholic fatty liver disease is very high. In the US, it's estimated to be 80 million. So it would be great to have a tool that would tell us which are those patients that are at higher risk that would definitely benefit from surveillance programs. For early detection, we talk about alpha-fetoprotein ultrasound that is relatively poor, its performance. Also for diagnosis and prognosis, we can improve. And certainly the second need that I think is more relevant is treatment response prediction. So how can we better assign treatment options based on the probability of the patient responding to that specific treatment? Now, there are only AFP is recommended for early detection and for selection of patients that will benefit from ramucirumab. Let's move again to the next, the second part of the presentation, where I will discuss in a little bit more detail what I think is going to be the role of liquid biopsy in the management of patients with HCC. For those of you who are not familiar with this concept, liquid biopsy entails the analysis of tumor components that are released to the bloodstream or any other biological fluid, but we'll focus on most of the people who's working with liquid biopsy in the blood. There are 3 major components from the tumor that are released to the bloodstream that can be isolated, can be quantified, and can be measured. One are the actual tumor cells, circulating tumor cells that are the biological substrate of distant metastases, along with lymphatic dissemination. Second component are CTDNA. These are fragments of DNA. When the tumor cells die, the DNA is chopped in fragments of approximately 150 base pairs, and this is released to the bloodstream, can be isolated and analyzed. And the third component are exosomes, which are small vesicles. The content is variable, but mostly they have small RNAs, and this is a system of communication between cells. So you can learn a lot about, understanding the content of these exosomes and how cells behave and how they communicate with each other. Now, the role of liquid biopsy in HCC has been studied for these 3 different components, and each one has its advantages and its problems, let's put it that way. So I wouldn't say that all of them work for everything, but certainly each one has its potential role in the management of patients with HCC. Now, when you draw a tube of blood and you centrifuge the sample, you get 3 components. the bottom, you have the red blood cells that now currently we're not using for anything. Then you have a thin white layer that is called the vesicle that has blood cells mostly, and then you have the plasma. Now, from this thin white layer is when you mostly will collect the circulating tumor cells, and you have the immune cells. So, as you know, immunotherapy is a big thing now in oncology, and everybody's just trying to identify biomarkers of response to immunotherapy. And I wonder if systemic readouts coming from immune cells in the blood could be useful. There's not much done in HCC so far, but I think it's a promising source of biomarkers. And in plasma, what you have is CTDNA and the vesicles that I mentioned before. For CTDNA, the concentration is very variable. For most of the cases, less than 10% of the DNA that is in the blood comes from the tumor. And this is probably an overestimation. It's likely less than 5 or even less than 3%. Because in the blood, you can also have fragments of DNA from other cells that die that are not tumoral. It's just normal turnaround of the cells. And you can find also there. So one of the main challenges is to figure out ways to discriminate the DNA in the blood that comes from the tumor from the DNA that comes from everywhere else, with a caveat that the percentage of DNA that comes from the tumor is generally lower than from the rest of the body. The average length of the fragments is between 140 and 170, and this is thought to be the part of DNA that is encapsulated in the nucleosome is very stable, and the half-life is between 50 to 120 minutes. And we know that based on studies that took DNA in patients with cancer before and after surgery. And when the tumor is completely removed, the concentration of the amount of tumoral DNA drops relatively rapidly. Now, if you go back to the natural history of HCC, I mentioned, remember that patients with cirrhosis are at high risk of developing hepatocellular carcinoma, and the risk is between 2 to 3% per year. And this was the rationale to include in these patients in surveillance programs. Now, let's put specific numbers to what this represents in the US, and this is data from the United States. There's approximately 600,000 cirrhotic patients in the US. If we assume that between 2 and 3% will develop HCC every year, we have around 18,000 new HCCs per year in these cirrhotic patients. Now, we know that the performance of the recommended tools for early HCC detection, that's ultrasound and AFP, has a sensitivity of 63%. So out of the 18,000, we'll be able to detect around 11,000 with the tools that we have, which leaves us with around 40% of the cases, a little bit over 6,600 patients that will not be detected because the tools are not good enough to identify these patients. Well, this is kind of the ideal scenario, but there's something else that needs to be computed here. And that's the proportion of patients with cirrhosis that are actually enrolled in a surveillance program. And that is very low. Recent estimates propose that only 25% of the patients that should be on surveillance are actually on surveillance. So if we compute that in this figure, we have the 600,000 cirrhotics, the 18,000 that we develop HCC every year. Now, only 25% of them will be enrolled in our surveillance programs. If we apply here the performance of the early detection tools, we have that only 15% of the total amount, so around 3,000 patients, will be actually detected, leaving a staggering 84% of the patients, 15,000 that would not be identified because either they're not enrolled in surveillance or because the tools are not good enough to detect early stage tumors. So here I have the two barriers for early HCC detection. We need to improve the accuracy of the tools, and we need to increase implementation of surveillance. And we wonder if by applying liquid biopsy, we hit both. So we have a better tool that is more accurate, and we have a method that is simpler, that does not imply the patient going to a hospital every 6 months to get an ultrasound, because you have to do that for years, and patients get tired, and there's significant attrition after, 4 or 5 years. Patients say, you know what, what's the point? And, every time I come here, the ultrasound is fine. Why should I do that? So if we had a method that is simpler than that, maybe a blood test, you go to a pharmacy, you ship it to central lab, and you get the result, may increase or decrease attrition for surveillance in these patients. Now, in terms of what is out there, what has been published for early detection using CTDNA. These are the two studies that have been, evaluating this in HCC. The first one is from China, almost 2,000 patients, case control. They use these markers, specific CPG sites detected on ctDNA. Sensitivity of the training set was 85 with specificity 94. The validation 83, specificity 90, which is outstanding. The second study was done in the US, in the Mayo Clinic. Very similar design, case control, also reported very high data in terms of sensitivity, around 80% for early stage. Now, both studies have the same problem. And the problem is that the majority of patients that they included, majority of patients with HCC that they included, have advanced stages. They're not at early stages. So when we talk about early detection, you need something that detects early tumor, because the most important objective, the aim of the surveillance program is to increase survival, not to detect more tumors, but to detect them at a point where you can do something about it, you can cure the patients. So these two studies have similar limitations, but I know that they're enrolling additional patients with much more earlier disease. We have been very interested in applying extracellular vesicle analysis for early HCC detection. The field of extracellular vesicles has evolved in recent years. There's a lot of new knowledge on basically what's the content of the vesicles, what's in there, what's the function of the vesicles, and something that came quite common in the majority of the studies. There's significant heterogeneity in these vesicles, in the content, in the functional, in the way you can isolate them and detect them. These the peer-to-peer studies in Cell, where they presented kind of a technical overview of different methods to isolate, analyze, and quantify cellular vesicles. And for instance, here you have different methods to isolate vesicles, and the components that can be detected using different methods are very variable, and that somehow could justify why some of the studies get not good reproducibility. So maybe the methods are just looking at things that are different. And the other thing that I find striking from this study was when they analyzed plasma vesicles, they found that a number of RNA products, and here you have it in green, are mapped to the genome. So there's kind of a black box of content in terms of potential functional, or potential raw as biomarkers that was available, and we thought that was interesting, and one of the reasons that we launched our study. So we did this study where we isolated vesicles using ultrasound isolation, which is a rather dirty way of isolating vesicles from healthy controls and patients with HCC. We did a number of studies to make sure that we were isolating vesicles, including Western blotting and electron microscopy, and immunolabeling and NTA tracking analysis. So again, we want to be sure that what we isolate would actually contain in the vesicles, and we were not getting significant noise from other components. We did a pilot study in 15 patients, 10 patients with HCC and 5 controls. We isolated the vesicles, and we did RNA-seq on the content of the vesicles, and we found a set of markers of small RNA sequences that were able to accurately discriminate patients from controls. Here, we selected 3 markers, small RNA clusters, that were significantly upregulated in our HCC cases compared to controls. And I want to underscore the fact that all of our cases are 85 HCCs or early stage HCC with a median tumor size of less than 3 centimeters. The patients, the controls, again, these are not healthy individuals. These are patients with chronic liver disease that are actually enrolled in a surveillance program. So this is the target population of surveillance. We had around 100 of them. Now, when you look at the performance of our 3 markers, here you have the performance, the predicted performance of ultrasound and AFP based on the meta-analysis that I told you. So 63% of sensitivity and around 83% of specificity. This is the curve of performance after cross-validation of our 3 markers. Here you have in yellow or in orange the performance of alpha-fetoprotein in our cohort, which was, better than we expected. And here you have the performance of our 3 markers plus alpha-fetoprotein. which outperform any of the two with a sensitivity of 86% and a specificity of 91%. And what we found also is that when you compare the expression of our 3 small RNA sequences with alpha-fetoprotein, they were not correlated, which makes sense to suggest that we're providing complementary information in terms of detection capacity for patients with early stage HCC. So in addition of early detection, the second clinical niche that I think that has the most future in terms of biomarker development is biomarkers of treatment response in patients with advanced stage HCC. So here you have a summary of the current approach for the management of patients with advanced HCC. The combination of atezo-bev that I mentioned before is the first line. They have sorafenib and lenvatinib. That was previously the first line, now it's considered either second line or first line for patients that cannot receive atezolizumab plus bevacizumab. And then you have third line options, regorafenib, cabozantinib, ramucirumab, nivolumab, or pembrolizumab. As I mentioned before, ramucirumab in patients that have high levels of AFP. Nivolumab was recently withdrawn from its initial approval under the accelerated program because, as you know, or probably you don't know, but the phase 3 trials that were designed to confirm the efficacy of nivolumab and pembrolizumab were negative, but the FDA decided to keep pembro and discontinue nivolumab in terms of accelerated approval. And there's also the combination of nivolumab plus ipilimumab, also under the accelerated program. So as I mentioned before, when you move down this algorithm, again, it's unclear what will be the criteria to decide which patients receive each of these therapies. And clinicians, most of them do is they select the treatment based on side effect profile that would be more favorable for a given patient, or on subanalysis. But as you can imagine, it's not the best way to select therapies for a given patient. And we know that patients that are treated with immunotherapies and respond, they do extremely well. So this is data from the phase 2 trial that was testing nivolumab in patients with advanced HCC. That was the basis for the accelerated approval. You have survival according to response. In the green, you have patients with complete or partial response. As you can see here, the percentage is not very high. It's around 16 to 18%, but they do extremely well. So the median survival, so the half of the patients that had a response on nivolumab lived more than a year. And that's unprecedented for HCC at this stage. So who are these patients, right? So is there a way that we can identify the patients that will respond? Or the other way around, which are the patients that do very poorly on these therapies? So we tried to assess that, and we did a study using gene expression on tissues from patients with early stage HCC. It was led by Dr. Lovett a while ago. And we found that around 25% of the patients have what we call the immune class, and they have an enrichment of signals that suggests activation of the immune system, and this can be subclassified in other classes, active and exhausted, but the bottom line is around 25% of the patients could be classified in this class. There were other group of tumors. You have here the immune class around 30%, and you have other group of tumors around 30% that lack any signal of immune activation. Not only genomically, but when you look at histologically of the tumors, there were no immune cells infiltrating those tumors. They were completely cold. And these are the ones that we call immune excluded, and they were enriched in mutations of the Wnt beta-catenin pathway. Since these patients are at early stages, and this was before the immunotherapies were available, we couldn't tell whether these classes could be used as biomarker of response to immune checkpoint inhibitors. But what we did is we compare our signatures with others that have been shown to be effective in other malignancies. There was a significant enrichment, so our signatures were very similar to the signatures that identify responders to these therapies in melanoma and lung cancer. Whether that is enough, probably not. So we need additional evidence to support the role of these signatures to identify patients that will respond to immune-based therapies. More recently, you have the paper on Journal of Hepatology that report on potential biomarkers of response from the phase 2 trial, the nivolumab trial that I was mentioning before. So they run a number of studies on tissue samples from these patients, gene signatures, and there were some p-values and some association, but for instance, when you look at the percentage of positive PD-1 cells, according to response, you have here complete response and partial response, stable disease, and progressing disease. There's some p-value, there's differences in terms of the percentage of PD-1 positive cells in the ones that respond to the ones that do not respond. But you can see here, despite you have a p-value, there's significant overlap between the groups, and you cannot use that in clinical practice. You need something that is much cleaner, much more clinically actionable to implement and to recommend in guidelines. So we started working on CTDNA, and the question was, the first question that we posed a couple of years ago is, can we detect mutations in CTDNA of patients with HCC? So we did this pilot study where we enrolled 8 patients and we did ultra deep targeted DNA sequence of all exon of a panel of 58 genes that we selected based on whether they were druggable or they were prevalently mutated in HCC based on tissue analysis. Out of the 8 patients, we also did multi-regional sampling in different regions of the primary tumor, and we found 21 mutations in tissue. We have per tissue and blood for this cohort. And out of the 21 mutations in tissue, 71% we could detect in blood. Now, we confirmed these mutations using digital droplet PCR. We're quite confident that we have a tool that at least detected 71% of the mutations. But this was kind of the old days, and while we were doing this study, then the new technology, including barcodes, was developed. And we thought, well, if we can increase the accuracy, we may do better to identify biomarkers. So we designed a second study where we enrolled patients with advanced HCC 85. These are the patients that are not biopsy regularly. Remember I mentioned about the fact that most patients can be diagnosed without the need of a biopsy. So for these patients, we don't have tissue to do biomarker analysis. So we need another source to identify potential biomarkers. So here in these 85 patients, some of them were treated with tyrosine kinase inhibitor, 24, some of them were treated with checkpoint inhibitors, but they were all at advanced stage. We reduced the size of the panel for technical reasons. Instead of 58, we did 25 genes, again, genes that are frequently mutated in HCC or genes of the Wnt pathway, because we wanted to test the hypothesis that I mentioned before that those tumors with mutation of the Wnt beta-catenin pathway tend to be immune excluded. So if that's the case, is that a correlate that would tell us that the patients that would not respond to nivolumab or immune checkpoint inhibitor? So that's why we changed a little bit the panel. And we also did type promoter with digital droplet PCR because it was kind of difficult, and it's always very difficult to identify type promoter mutations using NGS. So we kind of use a different assay to call third promoter mutations. So we use the SureSelect technology. The library size was 225, and here you have the details of the sequencing approach and how we call the mutations, and you can find more details on the actual paper. So this is a summary of the mutational landscape of our 85 HCC patients. I want to emphasize the fact that these are advanced HCC, advanced stage, which is different to the majority of patients that have been published in all the TCGAs and ICGC that were all using resection specimen, which is a different stage. The first thing that we know is that for the majority of genes that we know are prevalent in early HCC, they're also prevalent in advanced. The most frequently mutated gene was third promoter with 51%, and p53 30%, beta-catenin 17%, and as in early stages, beta-catenin and third promoter mutations are mutually exclusive. Sorry, beta-catenin and p53 mutations are mutually exclusive. As expected, when you look at survival or progression-free survival based on response rate, so the patients that were treated with mostly with immune checkpoint inhibitors that respond, they do significantly better than the ones that did not respond. And this was expected based on the previous data. And here you have the characteristics of our cohort, the 38 patients treated with checkpoint inhibitors, the 23 treated with tyrosine kinase inhibitor. The majority of tyrosine kinase inhibitors was seraphin, I have to say, more than 90%, and the majority of checkpoint inhibitors was nivolumab, again, more than 90%. Things to highlight is that the cause, the etiology of the liver disease was mostly hepatitis C virus infection, and they were evenly distributed between both cases, even probably a little bit more of alcohol-related HCC also in the ones that received nivolumab. Then you have the majority, as mentioned before, were patients with advanced HCC. They had multinodal disease in 80% of the cases. And also we had patients with extrahepatic spread in almost half of the two cohorts, with a median ASP of 420 in one case and 785, all of them above 400, which is the cutoff used to identify patients with aggressive tumors. So in summary, we're dealing with a cohort of patients with advanced tumors with aggressive tumors and relatively difficult to treat. Here are the results of the patients that were treated with tyrosine kinase inhibitor and the mutation analysis of these patients. We found that patients with mutations of genes belonging to the PI3K-mTOR pathway tend to do worse as opposed to the ones without these mutations in the patients receiving sorafenib or TKIs. This did not happen in patients treated with checkpoint inhibitors, suggesting that this could be a biomarker of lack of response, in this case, to sorafenib. It's interesting because a couple of years before we published this study, the group of Memorial Sloan Kettering, led by Jim Harden, they also found that mutations of patients with HCC and mutations in genes of the mTOR pathway, they didn't have also response to tyrosine kinase inhibitor. But the main difference between their study and our study is that they used tissue as opposed to us, that we use blood and CTDNA to call the mutations. When you look at the patients that received checkpoint inhibitors, nivolumab in our case, we found or we did not found that patients that had mutations in the Wnt pathway did worse with nivolumab. Actually, we had a couple of patients with mutations in beta-catenin, confirmed mutations, that had very good responses. We raised the question of whether, it's not only the fact that you need to have the immune cells within the tumor, but maybe there may be a role of nivolumab or checkpoint inhibitors to facilitate recruitment of immune cells within the tumor, and that way increase response to these therapies. There's another thing that we introduced in this paper that I think is by far the most valuable thing about liquid biopsy. And so most of the people would say, well, the number one benefit of liquid biopsy is that you don't need to do a tissue biopsy that has complications, that is invasive, that is annoying, and that, patients are much better doing a blood test than having to do a tissue biopsy. And that's true, and it's a major advantage. But to me, by far, the main advantage of liquid biopsy is you can do it sequentially. So if we assume, and we know for a fact, that tumors evolve over time, they change when they're exposed to different treatments, there's a process of clonal evolution of tumors, and we're pretty far away from studying that clonal evolution in real time in patients. So you think that it's difficult to get a tissue biopsy from an HCC patient? It's almost impossible to get 3, 4, 5 tissue biopsies from a patient, before it receives a treatment, after it receives a treatment, before progression, after progression. It's not going to happen. It's unrealistic. But you can certainly take one blood sample every time the patient comes to the clinic. So that's the game changer in my view, and that's why I think liquid biopsy will become mainstream sooner or later for the management or to better understand how tumor evolves and how tumor interacts with our therapies, with the immune system, in a way that we can use that information to derive biomarkers. We have 2 examples of a progressive disease and objective response in 3 patients, in this case, in our study, and also 2 patients 7 and 8 in the other part of the figure. And what you can see here is that the mutations change. It changed the binding fraction, the prevalence, and changed the actual mutations. And in one patient, you see that upon progression, there's an emergence of a 53 mutation that was not present at baseline. Well, you can say maybe it was present, you didn't detect it. It's possible, but it's conceivable to believe that this patient may have developed a mutation, over time with a new clone or with part of an evolution of emergence of a more fit clone. We have also worked on developing experimental models to study liquid biopsy, and this is a very easy approach. We inject subcutaneously in nude mice 2 cell lines that have different genetic makeup, trying to mirror, to mimic heterogeneity in humans, and they were treated with sorafenib or placebo. This is at the time was the drug that was more readily used for patients with HCC, and you can analyze the blood of these mice and detect mutations. In our case, we use these on called PCR, and it's kind of an easy way or a easy, feasible tool to study clonal dynamics and how a liquid biopsy, in this case CTDNA, can help us trace clonal composition over time, and when patients, or in this case when animals are exposed to different therapies. So just to summary the presentation, I would say that liver cancer is a major health issue, and we know that its incidence is increasing worldwide, in the US in particular, and mortality is also increasing. There are specific areas in HCC research where biomarkers are needed, and I would like to highlight two of them. Early detection. The tools that we have are very poor. They only detect 63% of the patients with early tumors. We need something that is better than that, because we're missing a lot of patients with early tumors that can be cured. And secondly, biomarkers are needed to tell us which is the best sequence of treatments the patient will receive to maximize efficacy, minimize toxicity. We know that mutation analysis of ctDNA identifies candidate biomarkers resistant to sorafenib. We show the data of mutations in genes of the mTOR PI3K signaling pathway. Whether that could improve in trial design is still to be studied, but certainly will help to facilitate the development of new biomarkers using ctDNA. I think there's no question that liquid biopsy has emerged as a promising and above all, a very convenient tool for biomarker development in HCC, and I would say in oncology in general. And there are a number of studies that show its power to detect, minimal residual disease and identify patients not only in HCC, but in colorectal cancer and lung cancer. With that, I want to thank people in the lab, former members of the lab that were leading these studies, particularly Johan Bonfelden, a postdoc, a very talented postdoc from Germany, he's back in Germany, that led the ctDNA study and the extracellular vesicle studies, along with Teresa Garcia-Lezama, that was also very talented in the lab working on the extracellular vesicles study. I thank you for your attention, and I'm happy to take any questions.
Thank you, Dr. Villanueva, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what were your criteria for curating the 25 gene panel used to profile ctDNA samples?
Yeah, so in the first study that we analyzed the ctDNA, the pilot, we used a panel of 58 genes, and we decided to scale it down first to reduce cost and to be more selective in the type of information that we're going to be achieving. So the main criteria were, first, we looked for prevalent genes, so prevalent genes that are mutated in HCC based on tissue studies. The top, let's say, 15 genes that have been reported to be mutated in HCC. Because we wanted to know, if the mutation profiling in advanced cases was similar or mirror a bit what has been reported in early stages. And the second is, one of the reasons that we conducted these studies, we want to confirm or we want to test the hypothesis that patients with mutations in beta-catenin did not respond to checkpoint inhibitors, specifically nivolumab. So we included a number of genes, 5 genes, that were part of the beta-catenin pathway, known to be mutated in HCC, beta-catenin, AXIN1, AXIN2, to specifically address that question. So those were kind of the two main criteria we follow to address that.
Okay, thank you. Next question. Currently, is it worth doing research work on the effect of sorafenib on hepatocellular sarcoma?
Yeah, that's a very interesting question. And I guess the reason for the question is, so 6 years ago, there was only sorafenib approved by the FDA, and now we have 9 drugs approved. So is there any role of sorafenib in the management of HCC? So it's worth to invest money and time to study the effects and the biomarkers to sorafenib? I think the answer is still still yes. Obviously not as dominant as it used to be. But if you think about it, in first line, the recommended therapy is atezo-bev. But in the trial and in clinical practice, patients that have varices in the esophagus, that's a complication of cirrhosis. You develop veins in the esophagus that can break. So these patients cannot be treated with this combination. This represents approximately 30% of the patients. Then these patients will be eligible to be treated with sorafenib or lenvatinib. So still sorafenib has a role in the clinical management of HCC. It's not as dominant as it used to be, but I think that it's still a niche to keep studying or getting additional information of the role and potential biomarkers of response to sorafenib.
Okay, thank you. Next question. Did you test other liquid biopsy samples like urine for your studies?
No, that's the short answer. And there's no specific reason, because there are studies showing that you can quantify some of these analytes in urine, and it's even easier for patients because you can think about home collection of samples. But for a number of reasons, we focus on blood, but certainly urine or even saliva are very interesting sources for running this type of analysis.
Okay, and next question. Excuse me. This is a bit of a technical question. With your 225 kbp probes being on the low end of what the SureSelect technology is designed for, did you have to optimize much to achieve a low off-target coverage?
Yeah, so I would say that in general, the whole technical process of generating the data was relatively straightforward. So there was not a specific need to, do a lot of troubleshooting to generate the data. I guess that, the main bottleneck on our end is that the starting material is 4 mL of plasma, which is kind of a little bit too much. We scale it down to 2 mL, but from that point, the centrifugation of the sample, the isolation of the DNA, and all the downstream analysis and data generation pipeline, we didn't have many issues, I have to say.
Okay, thank you. Next question. Did you detect any stromal fibroblast markers in the liquid biopsy projects you were involved, or are you aware of any published reports on this issue?
So we did not found any specific stromal markers, so I cannot tell you specific about that. I don't know of any specific project looking at liquid biopsy surrogates of the stromal component of the microenvironment, but so in addition to liquid biopsy, we've also worked in projects related to HCC evolution and into tumor heterogeneity and single cell sequencing, and it's amazing the dominance of the tumor microenvironment in all the studies, even when you look at functional heterogeneity at different parts of the primary tumor. So I wouldn't be surprised if it could be another potential source to detect or to derive biomarkers on tumor progression or response to therapy, specifically looking at the microenvironment.
Thank you. Next question. Do you see CTCs one day replacing AFP testing altogether?
I don't think so, but I will rephrase a little bit the question. Do I think CTDNA analysis replacing AFP? And to be a little bit more provocative, I see CTDNA analysis replacing AFP plus ultrasound in surveillance programs. Yeah, I do think so. Either CTDNA or extracellular vesicles, based on what has been published and our data, I think that they're very promising, not to outperform AFP, which, to be honest, is kind of sort of a relatively bad biomarker. Is to replace the whole thing, AFP plus ultrasound, and that's the objective, that's the aim. Simplify the whole surveillance effort and do a blood-only based test that is easy to implement, that is accurate, and that can help us increase early detection rates for patients with HCC.
Okay, next question. Is it possible CTDNA can identify important molecular markers that could be missed if another tissue biopsy is difficult to pursue?
So I'm not sure I understand the question, but if you're referring to the fact that, so in our analysis, we're very targeted in what we're looking for. So we have 25 genes, and you could argue, well, it's kind of a limited scope of what you're looking for, right? And it's true. So you don't have the coverage or the depth in analysis that you would get with whole exome sequencing when you do tissue, and you may be missing, information or genes that are mutated that could be relevant. That is true, but it's always a trade-off. I know there are some studies doing whole exome sequencing plasma. I think that there's some technical issues and may not be still mature, but certainly you can expand to larger, bigger gene panels, 400, 500 genes with very good results with the newest technology. So I think that still we're kind of focused on a small portion of the story of potential genetic alterations in HCC, but I found that by selecting that, at least we get some information without the need of doing a tissue biopsy.
Okay, thank you. Next question. Can you comment on your method of cfDNA extraction in typical yield?
Yeah, so as I mentioned before, one of the main not an issue, but something that we would like to improve is that the input amount of plasma that you need is relatively high, 4 mLs ideally, but then scale it down to 2 mLs. But the process is relatively straightforward. There are a couple of rounds of centrifugations, and then we use commercial available kits to extract the CF circulating free DNA. So the concentration that we obtain, and we did this both in plasma and in serum to see if the yield or the performance of mutation calling in these two compartments was different, and we didn't find any differences. So you can use either plasma or serum, at least for our study. The median concentration that we detected was around 10 nanograms per ml, which is low, but there are tools that can handle that low amount of DNA.
Okay. Next question, what is the treatment for liver cancer in advance?
So if you're referring to liver cancer at advanced stages, there are a number of systemic therapies that have been approved. Specifically, there are 8 drugs, compounds approved by the FDA. And that's essentially the mainstay without going into much details. If you're talking about prevention of HCC, so let's say that you have a patient with cirrhosis and you know that the patient is at increased risk of developing HCC. So why don't we give the patient something to prevent liver cancer development? So there has been a number of trials testing different compounds, vitamin A analogs, sorafenib was tested to prevent recurrence, vitamin K, retinoids, and none has proved to be effective to prevent HCC development. So there are no approved drugs in chemoprevention for HCC.
I see. Next question. What is the best way to stimulate mice hepatocellular carcinoma in a short time?
So if you're asking which is the best animal model in terms of, which is the one that more quickly will develop HCC, so obviously the fastest on our hands was the subcutaneous injection of cell lines, which is in immune mice, which is not the best for a number of reasons, but it's certainly very quick. The other model we've used, not for the liquid biopsy studies, but for other projects, is hydrodynamic tail vein injection of genetic elements that get delivered to the hepatocytes directly. And that's very convenient because you can, let's say, manipulate a number of genes. You can induce mutations, beta-catenin mutations, MYC overexpression, and you can get tumors in the liver, so orthotopic models, in 3 to 4 weeks, which is very, very quickly. When you go to chemical induced models, DEN, CCL4, those take significantly longer. So depending a little bit on what type of project that you're interested in, you can use the different approaches. But you can have models developing HCC as short as 3-4 weeks.
Thank you. Next question. What was the sequencing depth used in your CTDNA study and variant allele frequency detection limit achievable with the sequencing depth?
Yeah, so on average, the depth of sequencing was between 5,000 to 10,000x, depending on the two studies that we did. And the VAF of the detection limit was 0.1%. I have to say that for most of the mutations that we detected, the VAF was around 1%. Remember that I mentioned in the presentation that less than 10% of the DNA in the blood comes from the tumor. Well, you have outlier patients that the percentage is high, but the majority of the patients were around the 1%, which is what complicates a little bit the analysis and when you're working with ctDNA.
Okay, well, it looks like we have time for one more question. Did you use UMI to sequence cfDNA? And can you comment on the importance and necessity of using UMI?
So in the first study, the pilot study where we did the paired samples, tissue and blood from the 8 HCC patients, we didn't use UMI barcodes. And one of the problems that we have is that we have a significant number of false positive calls. So variants that we detected that were not true mutations, but sequencing errors. So with UMIs, you reduce that dramatically. And it was a big advantage in the second study where we did apply UMIs and the percentage of false positive calls was significantly lower. So I think that at this stage, it's very difficult to do a comprehensive study and get reliable information of the ctDNA space if we're not using barcodes or UMIs.
Thank you, Dr. Villanueva. Do you have any final comments for our audience?
No, I hope that you enjoy the presentation and you see that liquid biopsy certainly moving forward is a potential new tool for the management of patients with cancer in general and with HCC in particular. I think there's still some, additional problems that need to be sorted out, but I certainly see that's the future for the management of patients with HCC.
Thank you again, Dr. Villanueva, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Agilent, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.